Hello, welcome to Biology, the scientific study of life. More about the Mendel and the gene idea. Inheritance patterns are often more complex than predicted by simple Mendelian genetics. Notice this example shown the difficult or the complex of of inheritance. Incomplete dominance in a snapdragon color. When red snapdragons are crossed with white ones, the F1 hybrids have pink flower segregation of alleles into gametes of the F1 plants results in an F2 generation with 1 to 2 to 1 ratio for both genotype and phenotype. Superscripts indicate the alleles for flower color C to the power R and C to the power R for the red and C to the power W for the white. Notice also the pink. More about the extending Mendelian genetics for two or more genes. A pletropy. So far we have treated Mendelian inheritance as though each gene affects one phenotypic character. Most genes, however, have multiple phenotypic effects as property called pletropy from the Greek plenmor, which means, for example, pletropy alleles are responsible for the multiple symptoms associated with certain hereditary diseases in humans, such as cystic fibrosis, and scale cell disease. We will discuss it later. Considering the intricate molecular and cellular reactions responsible for an organism's development and physiology, it is not surprising that a single gene can affect a number of characteristics in an organism. Notice the determination of ABO blood group by multiple alleles. Notice the genotype I to the power capital I to the power A capital or A to the power capital I small. Notice the phenotype blood group A. Notice this blood cells. AI to the power B capital, I to the power B capital, or I to the power B capital I small. The phenotype blood group B, I to the power E, I to the power B. The phenotype blood is AB. I small, the phenotype blood group O. Extending Mendelian genetics for two or more genes, dominance relationships, multiple alleles, and pletropy all have to do with the effects of the alleles of a single gene. We now consider two situations in which two or more genes are involved in determining a particular phenotype. Epistasis. In epistasis, from the Greek for stopping a gene at one locus, alters the phenotypic expressions of a gene at a second locus. An example will help clarify this concept in mice and many other mammals. Black coat color is dominant to brown. Notice this figure, an example of epistasis. This Punnett square illustrates the genotypes and phenotypes predicted of 4 offsprings of matings between two black mice of genotypes B capital B small, C capital C small, and C capital C small gene, which is epistasis to the B capital B small gene. Notice that gene controls whether or not pigments of any color will be deposited in the hair. Notice. A simplified model for polygenic inheritance of skin color. According to this model, three separately inherited genes affect the darkness of the skin. The heterozygous individuals, A capital A small, B capital B small, C capital C small, notice that, represented by the two rectangles at the top of this figure, each carried three dark skin ovals, black circles, and three light skin ovals, open circles. The variations in genotype and skin color that can occur among offspring from a large number of hypothetical matings between these heterozygotes are shown above the graph. The x-axis represents the fraction of progeny with each skin color. The resulting histogram is smoothed into a bell-shaped curve by environmental factors that affect skin color. In greeting, notice that interrogating a Mendelian view of heredity and variation. The effect of environment or phenotype. Notice that the outcome of genotype lies within its norm of reaction, a phenotypic range that depends on the environment in which the genotype is expressed. For example, hydrogeny flowers and of the same genetic variety ranged in color from blue to the pink, depending on the acidity of the soil. Notice the variation. Thanks for listening.